Welcome you all. This is Dr. Megala, Associate Professor, Department of Genetic Engineering, SRM Institute of Science and Technology. Would like to present a video on qualitative analysis of monosaccharide, glucose from known sample, blank sample, and glucose from food source date sample. The video will be helpful as it covers the basic information on monosaccharide, principle involved in the analysis, reagent preparation, and test involved in screening for the presence or absence of glucose in known sample, food source, and blank sample. Monosaccharides belong to the classification of carbohydrate, and they were classified as simple sugars because they cannot be further hydrolyzed into any form of sugars. They can be further categorized based on number of carbon atoms such as triosis, tetrosis, pentosis, and hexosis. Also, they can be categorized based on functional groups as aldosis and ketosis. Qualitative analysis of monosaccharide glucose helps to check for the presence or absence of glucose in the given sample. For our experiment, we have taken 3 samples. One is known glucose sample, another is the blank sample, and the third one is the glucose-rich food source dates. Qualitative analysis of monosaccharide glucose involves the following test, which includes Molish test, Felling's test, Barfoot test, and Benedict's test. These were said to be the preliminary test, whereas phenylhydrazine test or ozone test is said to be the confirmatory test for the presence of monosaccharide glucose. The material required were glasswares, test tube holder, water bath, spatula, dropper, glucose and food sources, rich of glucose, microscope. Reagents required were Molish reagent, iodine reagent, Felling's reagent, Benedict's qualitative reagent, Barfoot reagent, Selvanov reagent, Bial's reagent, phenylhydrazine hydrochloride, sodium acetate, and glacial acetic acid. Molish test were the preliminary test, as I told you earlier. The reagent preparation for the Molish were prepared manually or you can get it commercially. The Molish test works on the principle based on dehydration of carbohydrate by sulfuric acid. Most of the sugars will be forming for fural or they produce hydroxymethyl for fural, which then condenses with alpha naphthol, resulting in the formation of a purple color compound. The procedure involved in Molish test is that take few ml of the given sample in the test tube and add 1 drop of Molish reagent and followed by which add 1 to 2 ml of concentrated sulfuric acid along the sides of the test tube and wait for the purple ring formation at the junction. So this is how you get a purple color ring at the junction if the sample contains glucose. If you get a brown color, this may be due to charring and you can dilute your sugar sample and repeat your analysis. So the next preliminary test is the Felling's reagent. And here is the preparation of the Felling's solution. You have 2 components in the reagent called Felling's A and Felling B. And mix equal volume of Felling A and B. This gives you Felling's reagent. The principle behind Felling's test is that the Felling's reagent reacts with the reducing sugar, that is glucose, on heating and reduces cupric ion to cuprous ion, producing a red color copper oxide. So the procedure involved in Felling's test is that take 2 ml of the given sample solution to which add equal volume of Felling's reagent and place it in the boiling water bath and observe for the color change. there will be change in the color from green, yellow to red precipitate. So the color changes from green to brick red is due to the concentration of glucose found in the sample. So the next test following Fellings is the Barfoot test. The preparation for the Barfoot is given here. The principle of Barfoot test is that the reducing sugar will reduce the copper to acetate to copper 1 oxide, and which forms a brick red precipitate. The procedure involved in Barfoot test is that take around 1 to 2 ml of the Barfoot reagent and add equal volume of the sample given to you and allow to boil by placing it in the boiling water bath for 5 minutes and check for the presence of a red precipitate at the bottom of the tube as it is shown here, or the red precipitate will be formed along the sides of the test tube. This confirms the presence of glucose. So following Barfoot test is the Benedict's test. Here is the reagent preparation for the Benedict's test, which involves solution 1 and solution 2. Benedict's solution reacts with the reducing sugar glucose on heating and reduces cupric ion to cuprous ion, producing a precipitate of red copper oxide. The procedure involved in Benedict's test is that take 2 ml of the known sample given to you or unknown sample or the food sample given to you and add equal amount of Benedict's reagent and place it in boiling water bath for a few minutes and observe for the color changes. If the color change is from green, yellow to brick red, indicates the presence of the glucose found in the sample. The color change from green to brick red indicates the concentration of the sugar found in the sample. Hence, Benedict's test is said to be the semi-quantitative test to indicate the approximate concentration of the sugar found in the sample. Phenyl hydrazine test or osazone test is a confirmatory test for any sugar given to you. The given sugar monosaccharide is confirmed with this osazone test. The reagent involved in preparing the osazone test is phenyl hydrazine hydrochloride you required sodium acetate, you required glacial acetic acid. Osazone formation involves hydrazone formation at C1 if it is aldose and C2 if it is a keto sugar. And there will be oxidation at C1 or C2. And there is no further oxidation from C3 carbon. The procedure involved in phenyl hydrazine test is that take around 5 ml of the sample given to you, add 3 ml of phenyl hydrazine hydrochloride solution and keep it in the boiling water bath and note down the time taken for formation of the yellow osazone crystals. The given sugar, if it is glucose, the crystal formed will be within 4 to 5 minutes, and you would observe an yellow needle-shaped crystals as formed here. there are few tests which were turning negative for monosaccharide glucose. Those tests were Bial's test, Selvinov test, and iodine test. Bial's test will be negative for glucose and positive for pentose sugar. The pentose sugar is ribose sugar. Selvinov test will answer for ketosis and not for glucose. Iodine test is negative for glucose. It will be positive for polysaccharide. Hope the video is helpful for the viewers who are interested in qualitative analysis of monosaccharide glucose. Thanks for watching our video.